Sabeti's lab at the Broad Institute. I really appreciate you all taking the time out of your summers to listen to me discuss our work, especially those of you in Europe who are like enjoying one of your precious days off from the absolute madness of European football. Um, Before I get started, I want to take a second to really recognize the roles of three PIs and their labs in making all of this work a reality. Um, in addition to Pardis, Steve, and Ryan being brilliant scientists and mentors individually, their ability to really harmonize efforts across three separate institutions has been an irreplaceable asset, both to this project and also to my training. Um, I also want to take a second to highlight Rodrigo Castro, who is a brilliant postdoc in Ryan Tui's lab at JAX. He co-led this project with me from its earliest stages to where it is today, and his fingerprints are really over all over everything in this work. Um, so personally, the core biological question that first brought me to genomics and then eventually to machine learning is a desire to understand how genetic variation between individuals leads to the manifestation of different traits and diseases. Uh, and really unlocking this genotype to phenotype connection remains one of the main motivations that continues to drive the genomics field. So the past two decades have seen a rapid explosion in the number of genetic variants found to be associated with human traits, courtesy of genome-wide association studies. And by like 2019, this particular catalog had accrued over 270,000 genetic associations. However, despite this staggering number of revealed associations, only a minuscule fraction have been pinpointed to a specific genetic and molecular cause. So really, without a strategy to efficiently sort through all this information, we're going to fall short of a complete understanding of how this variation impacts human biology. And a principal discovery made by sequencing the human genome is that less than 2% codes for proteins. And in the years since, we've started to appreciate that as much as 21% of the rest of the genome is packed with non-coding regulatory elements. These cis-regulatory elements, or CREs, are the DNA sequences that encode the rules governing gene expression. And so currently, our limited knowledge of grammar governing CRE function makes it difficult to interpret the majority of genetic associations. While protein coding variation tends to be easy to interpret and has dramatic consequence, it's rare in comparison to non-coding variation. So a fundamental hurdle in interpreting genetic variants is that we don't fully comprehend the rules governing CRE function. For example, in this plot summarizes all known genetic associations with inflammatory bowel disease. On the left side of the plot, we see a few large effect protein coding variants in red. And while important, these variants are rare in the population, as indicated by the y-axis. Conversely, on the right side of the plot, it's packed with non-coding variants in blue. And while each of these non-coding variants are of small effect, there are a lot of them, and they occur very frequently in the population, and thus on aggregate are of great importance in our understanding of IBD as a whole. And while this may seem daunting, as I'm sure most people here or all people here appreciate, the machine learning community has made tremendous strides in harnessing the vast amounts of data from sources like ENCODE to build models that can predict the biological impact of these variants on chromatin. This was in part fueled by two landmark papers in 2015-16 that brought deep learning to bear on this problem for the first time. So these papers, I'm also highlighting them because they served as our jumping off point when we first started kicking around ideas in like 2019. I'm sure, as everyone is also aware, the field continues to evolve at blistering pace. And now people are producing models that utilize more and more genetic context and can learn a more complete representation of gene regulation and indeed transcription as a whole. So today, there are a number of really exciting cutting edge tools that we can use to read the genome and interpret the function of genetic variation. However, perhaps one of the ultimate goals in genomics is not only to understand the regulatory code, but also to write it. And for me, this also brings to mind a quote from Richard Feynman, what I cannot create, I do not understand. And so hopefully, through learning how to write genetic code, we will be able to truly evaluate our proficiency in regulatory biology. And hopefully, as these technologies mature over the next few years, we'll also start to be able to design elements that control gene expression for the benefit of research and therapeutics. And there are clearly other groups that share this vision. For example, the ARTS and SARC groups have really elegantly shown that deep learning models can guide the design of cell and tissue specific regulatory elements in Drosophila. And I encourage everyone here, if they have not, to take a look at these two papers. So we took a slightly different approach, and we were really interested in like testing the reliability of machine-guided design at scale. And so one of the tools available in our group and sort of the ecosystem of the TUI, Sabeti, and Riley labs are this tool known as massively parallel reporter assays. And they're a technology that enable us to measure the regulatory potential of hundreds of thousands of fragments of DNA at scale. So we start this process by designing and synthesizing a DNA library composed of hundreds of thousands of short 200 base pair oligos that we can then barcode into an oligo library and clone into a plasmid with a reporter gene. We then transfect cells with these plasmid libraries and isolate mRNA from the transfected cells and use sequencing to count the barcodes in the mRNA and clone plasmid libraries. And by calculating a log2 fold ratio between barcode representation in the mRNA isolate and in the plasmid library, we can quantify the impact of individual oligos on transcription. And these experiments are really cool because we can explicitly control the nucleotide sequences in the starting oligo library. And thus we can use MPRA to test various hypotheses at scale. For example, by synthesizing pairs of oligos that correspond to a reference and alternative allele of a CRE, we can quantify variant effects empirically by MPRA, measuring the difference in activities of the reference and alternate alleles. Alternatively, if we're interested in exhaustively testing the regulatory potential of sequences in a bounded genomic locus, we can design oligos that tile a genomic sequence at a locus and test their activity in a reporter assay and generate sort of a signal track that is reminiscent of one of the many other genomic signal tracks that you might be used to seeing. But one of the drawbacks of using MPRAs is while they are fairly scalable, they're not infinitely scalable. And so it would be really great if for additional experiments, if we could build good high quality models of this data. One of the other reasons that we were really interested in building a MPRA model directly is because there's some evidence that that's been generated from like particular cell lines and cell types that sometimes MPRA can be have a fairly low correlation to like traditional signals of enhancer activity. For example, this experiment from Paul Kavari's lab at Stanford generated MPRA data, ATAC data, and H3K27 acetylation chip from primary human keratinocytes and found a really modest correlation between MPRA signals and these two like functional genomics signals. We ourselves sort of kicked around trying to figure out what the correlation between functional genomic signals and MPRA would be. And we did this by looking at informer predictions and comparing them to our MPRA results. And while we didn't find correlations as low as that I'd shown in the human keratinocytes, we still find that MPRA expression generally has modest correlation to like a range of cell type specific features that are predicted by Informer. So then all of this motivated us to build a model directly from data using MPRA. And we were drawn to just building a convolutional neural network like Bassett because one, our MPRAs are testing the function of small fixed length elements, only 200 tokens long, and this is pretty consistent with the input requirements on Bassett. And two, we were planning on using the model with downstream applications that involve iterative model evaluations, and we expected to just need to potentially pan the model billions of times. So we wanted something that would be like computationally efficient. So we decided to adapt Bassett, a model which broadly predicts chromatin accessibility for 600 nucleotide sequences in 100 cell types, and we adapted it into what we call Malinois, a model focused on making MPRA predictions in 3 cell lines. So here's a schematic of our like final Malinois model. Without spending too much time here, the key tweak from Bassett is that we used a branched structure for the linear layers. And we just found through like experimentation and just in practice using this like branched architecture where each branch generates a prediction for each cell type, improved predictive performance. So then we trained this model on a large MPRA data set generated in the lab. It was approximately 400,000 pairs of variant of reference and alternative alleles that were tested in K562, HepG2, and SCANSH. And these cell lines are representative of erythroid progenitors, hepatocellular carcinoma, and a neuroblastoma, respectively. And so once we trained our model, we validated it on holdout sequences and find generally that Malinois was able to make accurate predictions and holdout test sets, generally having Pearson's R's of 0.88 or better. And this overall compares pretty favorably to the experimental reproducibility that we've seen. On the bottom row, I have an example of, I think, 20,000 or so sequences that are shared between 2 different oligo pools that went through the MPRA process several months apart. And like I said, while MPRA is a powerful technique, it doesn't have the throughput to generate truly genome-wide measurements the same way that DHS, ATAC, or ChIP-seq can. But we can make dense MPRA measurements limited to a megabase of the genome at a time, which is what we did at the GATA locus. And we find that sort of a segment of genome-wide MPRA predictions that we generated using Malinois correspond really well to the results of an empirical tiling assay with like a high locus-wide correlation. So once we built confidence that we could reliably read DNA sequences to predict regulatory activity, we next wanted to test if the rules learned by the model would be effectively used to write new regulatory code. Success here would both show that our models are generalizable and provide a blueprint to apply these experimental and computational techniques to synthetic biology more broadly. For this particular study, our goal is to design 200 nucleotide oligos that drive transcription in 1 targeted cell line while silencing transcription in 2 off-target cell lines. So as we consider measuring and traversing the functional topology of the landscape of possible 200 MERS, it's important to note that global optimization is not feasible. The total number of 200 nucleotide combinations is far greater than the number of atoms in the observable universe. And so to address this, we used some clever iterative sequence optimization algorithms, and sort of unified them all into like a single computational framework. And very generally, we typically start this optimization process at random sequences of unknown function, and then a batch of sequences is passed to Malinois to obtain predictions. And the predictions are passed to an objective function that induces a topology over the search space that reflects our design goals. In our case, regions of high cell type specificity. And then sequences are modified based on an update rule, depending on the particular algorithm we're using. At which point we can start the cycle over again until we're satisfied with the predicted function of the sequences and are prepared to test them empirically. So we also nominated natural sequences to serve as a baseline to compare the activities of our designed sequences. First, we selected regions of cell-type specific open chromatin as measured empirically by DNase hypersensitivity sequencing. We selected 4,000 of these sites corresponding to each of the 3 cell lines studied that were also supported by H3K27 acetylation to delineate enhancer activity. We next revisited our genome-wide Malinois scan, where we chunked the human genome into a 200 nucleotide sliding window with 50 nucleotide steps, and used Malinois to predict cell type specific activity for each of the tiles. And we picked the top 4,000 most specific, predicted to be most specifically active for each cell type. So then we constructed a fairly large, well, maybe modest at this point, as MPRAs continue to increase in throughput. So we generated this library to compare, I think, 51,000 synthetic sequences to 24,000 natural sequences, and also used about 1,000 technical controls. And we had a really talented undergraduate take charge of doing this experiment, and she really was the champion of making this experiment happen. So interestingly, like before we look at the results, one thing that we did was we tried to understand the semantic differences between natural and synthetic elements, and we did a fair amount of this using techniques such as topic modeling. One alternative analysis that another group did that I also found fairly compelling was just to look at k-mer content. And so in this plot taken from this preprint, this group compared sort of k-mer content of natural sequences in our study in red, and compared it to random elements that they just generated, and the synthetic elements that we also generated in green, orange, and blue. And you can sort of see that even just looking at k-mers, the synthetic elements are very different from the natural elements. And one of the other cool things that this group did is they applied a language model of human DNA sequence that I'm sure everyone here is familiar with, and looked at the likelihood value and looked at the log likelihood that the model generated for the natural elements, random elements, and the synthetic elements in our study. And they found this like striking separation in log likelihood between the native elements or the natural elements and the synthetic elements. And so to this particular sequence model, all of the synthetic elements look about as naturally human as purely random sequence, which I thought was pretty cool. Um, so basically, like we went into this with like no real like expectation on how well it was going to go. Um, so here's the distribution of Malinois predictions for CRE activity in each cell type, separated top, middle, and bottom by the intended cell type target of the CRE. Perhaps unsurprisingly, Malinois predicts that synthetic elements that it had a hand in designing on the right have the most cell type specific activity. And another notable observation is that the elements that we identified from cell type specific chromatin accessibility are just overall and generally predicted to be poor drivers of cell specific activity on the left. So whenever we tested these sequences empirically, we found the MPRA measurements to be highly correlated with the predictions, achieving an average correlation of like 0.86 across cell types. Both groups nominated by Malinois outperform DHS in generating cell type specific elements. So in order to like better visualize this as one summary statistic, we used a simple function to quantify the specificity of each element. So for each sequence, we can calculate what we call min gap, or the minimum gap between the targeted activity in the targeted cell type versus the maximum off target. So we calculated this value just as a clean summary of cell type specificity. And when we use this metric, we find that the synthetic elements consistently outperform both natural groups. And we also find that the natural sequences identified by Malinois do indeed outperform the DHS-derived elements as well. So we also relied on a geometric approach to determine cell type specificity. So these propeller plots are our way of projecting this three-dimensional data onto a 2D coordinate system in a way that highlights cell type specific activity. So for each sequence, So for each sequence, the difference in the maximum and minimum activity across cell types is the radial distance of each point, while the angle is set by the ratio between the maximum and medium activity across cell types. And so we have the schematic on the top that sort of gives you a sense for where points of different activity across the three cell types will land in this propeller plot. So highly specific sequences will land on one of the three axles of this propeller plot, while uniformly active sequences or uniformly inactive sequences will sort of collapse to the middle. And what we consider the strongest hits will be far away from the center and tightly hugging a particular axis corresponding to a cell type. So when we compare sequences nominated by Malinois, we can see the difference in separation. We can see a strong difference in separation between compared to sequences nominated using DHS. And we can also observe that the synthetic group has even more sequences than the two natural categories that are sort of adhering to this criteria for like high specificity. When we dug in to these sequences to really understand why they were cell type specific, we found like 36 enriched core motifs in our data set. And we found basically motifs that you would expect to be highly associated with to be highly enriched in sequences that were targeting a particular cell type. For example, like a clear example of this is K562 optimized sequences are highly enriched for the GATA motif. And this makes sense because GATA is an essential gene in K562 and is important for erythroid differentiation. Another clear example of transcription factor binding motifs that make sense are the HNFs. So we find HNF1B and 4A motifs to be highly enriched in elements that are specific for HepG2. I would say the story for me is a little bit more complex with SK and SH, so I'm not going to go too deeply into that. So after gaining some semantic insight into why and how synthetic sequences were being designed, we decided to test a handful of elements in vivo. And because we would only be able to test like very few sequences in vivo, we took further steps beyond looking at the sort of maximally MPRA measured specific sequences to further prioritize the elements that we would use for experimentation. We also focused on elements specific for HepG2 and SKNSH, liver and neuronal respectively, to make it easier to isolate and image transgene expression because we weren't really sure how sequences that may be specific for a blood cell, how easy that would be to image and follow in an animal. So we prioritize synthetic elements with both high empirical MPRA specificity, and we also used Informer to make predictions of tissue-specific specificity and used that in addition to help us prioritize sequences. These prioritized sequences were then cloned into a GFP vector for zebrafish and a LAXI reporter for mouse, and used a minimal promoter to sort of mimic the in vitro experimental design. And once these constructs were generated, they were injected into embryos using random integration for zebrafish and targeted H11 integration for mouse. So as I just mentioned, part of the prioritization included using Informer to simulate the synthetic elements, the impact of the synthetic elements on a transgene in different tissues. So here we simulated transgene insertion at the H11 locus in mouse, and we can predict chromatin accessibility at the transgene with and without the synthetic CRE and measure the delta in signal to sort of get an idea of how the element will behave. We then aggregated predicted enhancer signals in mouse for 10 different tissues into a single score for each tissue. Of these 10 different tissues that we generated predictions for, we believe that K562, HepG2, and SKNSH are sort of analogous to spleen, liver, and neurons, respectively. And so in the same way that we summarized MPRA signal specificity before, we can use min-gap again on these tissue-specific scores. And while imperfect, we found that these tissue-specific min-gap predictions on the y-axis are fairly well correlated to the MPRA measured min-gap values on the x-axis. And we reason that sequences supported by both MPRA and the appropriate chromatin signatures in the target tissues that we expected to see them would be the most likely to validate in vivo. So we selected sequences from the top right-hand side of these plots, and then did some additional curation based on motif content and nucleotide level contribution scores. In the end, we selected 3 liver targeting and 3 neuron targeting sequences for additional experiments. And we first did these experiments in zebrafish. So first, I'm showing you a whole mount of 5-day post-fertilization zebrafish embryo. On the left-hand side is bright field image of the fish. In the center is a GFP image. And on the right is a merge. So this first set of images is using a control reporter, and you can see basically no ectopic expression. we then put uh, we then put a particular HepG2 specific we then put a reporter with the HepG2 specific enhancer into the fish, and then we can see here like 5 examples where we're getting positive GFP expression in the liver of these fish. Um, and we quantified just to give you an idea of how reliable this is. Whenever we inject this particular HepG2 specific reporter into animals, we found nearly 30 like liver found approximately 30 fish with GFP expression in the liver and about 10 that were negative. Um, so here I'm showing another synthetic enhancer, this time specific for SKNSH. On the left side, we have bright field, and in the middle this time we have merge. And we can see really kind of like really nice expression in the neurons of these animals. And whenever we zoom in to this particular box, we can see that the cells that are glowing have are, I guess, anatomically neurons from sort of this the GFP in the cell bodies and being expressed in the axons, um, projecting from the brain and continuing down the trunk of the animal. Uh, so then we took that, so the SKNSH specific element that I just showed you, we then took that element that drove expression in zebrafish and asked, would it drive expression in mouse as well? So just to be clear, we thought this was going to be a hard task and was not one that we necessarily expected to be successful. We tried 2 different SKNSH specific elements for this experiment, and actually only one of them did work. But here is an example of a day 14.5 mouse embryo where we've hooked up the SKNSH specific enhancer to a LAC-Z reporter. And sort of we can see this like clear blue staining in the head of the animal. And so whenever we grow up these animals and to 5 weeks postnatal, we can also dissect the brains out and better understand where the expression is specifically in the brain. And so here I'm showing, I think, a mid-sagittal view of the brain. So like cut down the middle with the cortex on the top. And so on the top is a brain taken from a mouse with a control transgene, and on the bottom is a mouse that has a transgene containing an SKNSH specific enhancer. And you can see very clearly strong marking restricted specifically to the deeper layers of the cortex. Whenever we do some whole organ qPCR on these animals, we find that animals containing the synthetic enhancer have significantly higher RNA expression in the brains versus animals that have a control transgene, whereas in liver and spleen, the difference in RNA expression is not significantly different between the synthetic element containing animals and the control animals. So we also did some further image analysis in collaboration with the Joy Lab at JAX. So here I'm showing images taken from layer 6 of a negative control. In these samples, we stained we stained with neuron, which is a marker for neurons, IBA1, which is a marker for microglia, and GFAP, which is a marker for astrocytes. And we also have LAC-Z, which is the signal, which is our transgene signal. We find that in negative control animals that there isn't any LAC-Z expression in these slices. Whereas if we look at the animals with the synthetic neuronal CRE, we can see that there is LAC-Z expression, and it seems to overlap with a subset of cells that express the neural marker, but not the microglial marker or the astrocyte marker. So I think this is suggestive of this element having some sort of subcellular specificity that we did not necessarily intend for, and don't really have a clear answer to why is happening right now, but is nonetheless fairly interesting to us. So all of this work you can find on bioRxiv currently. We have a preprint up, and this is the title. I think in addition to what's in this preprint, I've shown you some work that we generated or some data that we generated during revisions with the help of these additional people. And I wanted to take a moment to really acknowledge everybody who has been part of this project across, I guess at this point, 4 different labs. You know, it has been an absolute honor to work with all of these people. I think it's not often that you get to work with so closely with so many scientists and have like a great experience with all of them. So, you know, that's definitely been awesome for me. Yeah. So, any questions?